Okay, I'm here with another video. Revision. This one isn't so late, so hopefully I should be able to get going and get it a bit longer, because obviously the recent ones have been only 30 minutes or so. This one hopefully will be a bit longer than that. So I have some biology notes up here. I don't know if that's what I want to get along with. Yeah, I was looking at, we were looking at, organic in the periodic table here. So I have a few things to consider. What I want to do. I think going over biology is the way to go. Um, so I will go and look for some resources that work here. But yeah, basically, and then we can get right into it. Okay, so I have some notes here on photosynthesis. So I'm gonna read through them. Photosynthesis is a reaction in which light energy is used to produce glucose in plants. This process requires water and carbon. Pretty basic here. Carbon dioxide, sorry. I don't know why I said that. With the products being glucose and oxygen, of course. So, of course, the equation there would be H2O plus CO2 with light produces C6H6O6 plus O2. There are two stages of photosynthesis. These are the light-dependent stage and the light-independent stage. The rate of photosynthesis is determined by carbon dioxide uh, concentration, light intensity, as well as temperature. Chloroplasts are the site of photosynthesis and are adapted to photosynthesis in the following ways. Contains stacks of thylakoid membranes called grana, which provide a large surface area for the attachment of chlorophyll, electrons, and enzymes. A network of proteins in the grana hold the chlorophyll in a very specific manner to absorb the maximum of light. Yes. The grana there, granum, and the individual thylakoids, and I believe these bits in between are called lamellae. A bit extra stuff there that you don't actually need to know, but maybe it could come up. I don't know. A network of proteins in the grana holds chlorophyll in a very specific manner to absorb maximum amount of light. A granule membrane has ATP synthase channels allowing ATP to be synthesized as well as being selectively permeable, allowing the establishment of a proton gradient. Chloroplasts containing DNA and ribosomes contain DNA and ribosomes, allowing them to synthesize proteins needed in the light-dependent reaction. Light-dependent reaction. Photons of light hit chlorophyll molecules in photosystem 2, causing the electrons to become excited. So this is the, oh wait. Why are we doing the light dependent first? Does that usually come first? I feel like I learned the light independent reaction first. Because that has the, what do you call it? Calvin cycle. But this is okay. Yeah, so. What is the light hit chlorophyll molecule cause electrons to become excited? This is called photoionization. The charge separation from this drives the process of photolysis. Photolysis is the splitting of water with light. One molecule of water requires 4 photons of light to split. Okay, in order to split, it produces one molecule of oxygen, 4 protons, and 4 electrons. The oxygen either naturally diffuses out through the stomata or is used in a respiration. Interesting. The 4 electrons replace those lost from the chlorophyll. Whilst the protons move into the stroma. later creating a proton gradient. Okay, that's important. The excited electrons then move down to a series of protein complexes. At one of these complexes, energy from the electron is used to pump 4 protons from the stroma to the thylakoid space. Okay. The electron then moves down the chain further to photosystem 1. Here, more photons of light are absorbed, causing the electron to move back up to a high energy level. When does it go down? There. Kind of missed that somehow, even though I just read it. The electron then moves along the chain to another complex where the electron combines with a proton to form a hydrogen atom. This is then used to reduce NADP, forming reduced NADP. Okay, perfect. Okay, where were we? The pumping of protons across membranes mean that there's now a greater concentration of protons in the thylakoid space than the stroma. As a result, the proton gradient forms with a high concentration in the thylakoid space and a low concentration in the stroma. The protons move across the membrane by diffusion through a protein known as a stalked particle. Not sure that is something that I have covered. This is AQA spec, so not sure why I haven't heard of that, but the movement of these protons drive the process of photophosphorylation. The enzyme ATP synthase phosphorylates ATP from ADP and PI. Carbon dioxide fixation. Carbon dioxide that is diffused in through the stomata is fixed with ribulose biphosphate IBP in a process known as carboxylation. The enzyme rubisco is needed in order to do this. A 6-carbon sugar is formed first. However, this is very unstable and therefore forms 2 molecules of glycerate 3-phosphate. Sorry, I didn't really get any of that. Carbon dioxide that is diffused in through the stomata is fixed with a ribulose biphosphate. I remember this. This is just the carbon cycle. Okay. The process known as carboxylation. The enzyme rubisco is needed in order to do this. A 6-carbon sugar is formed first. However, this is very unstable and therefore forms 2 molecules. I think getting up the Calvin cycle might genuinely be easier. Not Calvin Klein. Cycle. Here we go. Or this. These are just as good. 3 carbon dioxides, 1 rubisco forms. That's not as easy to interpret the 6 carbon molecule. Splits into 2 3 carbon molecules, which is ribulose. Nope. Which is PGA. Pen. Where are the names? Where are the names? There is no names in any of these diagrams. Phosphoglycerate. That's not important. Glyceraldehyde 3-phosphate. I think this is a different uh, spec. Where is my own spec? Is there a diagram in the summary notes? No. Yay. Reduction phase. There are 2 molecules of glycerate 3-phosphate contain a COH group and therefore an acid. The reducing power of reduced NADP therefore reduces the glycerate 3-phosphate with energy being provided by ATP. This therefore forms 2 molecule triphosphate. All of the NADP from the light dependent reaction has now been used with only some kind of ATP being used. So yeah, so glycerate 3-phosphate, that's the name of the 3-carbon compound there. The reducing power of reduced NADP therefore reduces the glycerate 3-phosphate with energy being provided by ATP, so therefore forms two molecules of triose phosphate. Yes, GP to TP. All of the NADP from the nitrogen has now been used with only some. I thought it was NADPH. From the nitrogen has now been used with only some of the ATP being used, which is now ADP plus PI. Brilliante. Regeneration of RUBP. 5 molecules of triose phosphate are used in order to regenerate 3 molecules of ribulose biphosphate. Along with the CO2, or what? Wait a minute. Wait a goddamn minute. 5? How does that make this? Oh, IBP is a 5-carbon compound. Oh, I see. So it's the ratio works then. Does it? Does it even work? 5 go to 3? Why does that work then? Why does 5 going to 3 work? Because we have 25. Oh, we have 15, 15. Oh, it does work. Oh, it does work. My bad. It'd be easier just to say that 1/5 and then you just say 1 IBV. Yeah. It makes sense, at least, so I won't fret on it. Where were we? Yeah. Yeah, this is wait, why is it 2? So it was 1. So it was 1/6. Fructose is a disaccharide. No, it's not. So what are we doing here? How have we got 7? Huh? Why have we got 2? We have 7 molecules of triphosphate here. Why do we have 7 molecules of triphosphate? Why do we have 7 molecules? Yeah, this, I think it's just been confusing because it is just 1. It's just 1. 2 total to make it. So I guess, oh, it's just saying it's 2 total that is required. That's just silly of me. Alright. Respiration is the next one, but I think we kind of managed to cover everything we need there, I guess. I mean, everything else I guess is covered in other topics, I suppose. So I guess now I'm just going to speed run through some questions. Don't really want this video to be too long. I have the daily to be doing, so yeah, there's that. And yeah, so it's 5 marker, but let's just break this down. So describe the way in which ATP and reduced NADP are produced in the light-dependent reaction photosynthesis. So that's the first part. We didn't go over that too long ago. ATP is produced by, okay, so there's the proton gradient across the ATP synthase enzyme, which catalyzes the reaction of ADP and inorganic phosphates to produce the ATP. For that proton gradient coming from the Comes from the photolysis of water. The NADP is produced by, well, it's reduced when hydrogen gas reduces it after being produced when electrons fuse with the protons that have diffused across the membrane through the channel, and then it becomes, yeah, and the electrons kind of bond with it and reduce it, hydrogen gas, and then it reduces NADP to produce NADPH. That's my understanding. Let's check the answers here. If we have answers here. We can have a PDF for the questions with the answers. I wonder if I go down, I'll find them. No answers. Where are the answers then? It's not what I want to look at. Oh, here we go. Okay, here we have one AI. Yeah, we said this through the, well, I didn't actually mention the photosystems, but, yeah. Energy is released. ATP formed from energy is released. But you have to say in context, formed from ATP and PI because of the ATP synthase, no? Where does it say ATP? Yeah, here, this. Is it not this? It is this. No? It's just kind of a wacky mark scheme, but I think I got at least 4 there in my explanation. And a full marker to follow. Explain how ATP and reduced NADP are used in a light-independent reaction of photosynthesis. So ATP is used first. Oh no. This is going to be rough. ATP is used first in the... I know it's used to reproduce the ribulose biphosphate or whatever. The RUBP. That wouldn't be ribulose biphosphate, would it? Why is it RUBP? Oh, ribulose. Yeah, okay. So yeah, it's used in that. That's one thing. And reduced NADP and ATP are also used in, which bit is it? It's a shame I've briefly forgotten. If we just check our diagram, we should have. Oh, we don't want to open that. We just want to open this a bit bigger. So yeah, here and here. So it's used from GP. Where's the TP? Oh, here's the TP. So it's used to go from TP to GP, or GP to TP. She didn't mention triphosphate here, so I don't know what that one is. Yeah, this is the one I'm used to. So yeah, the ATP and NADPH are used to convert 2-glycerate 3-phosphate GP to TP. GP converted to triphosphate, that's what it is. This involves reduction. For hydrogens, ATP supplies energy. Okay. Phosphate from ATP. Production of IUBP. Yep. Okay. To say phosphorylation or not? I hope not. So things to remember there are phosphorylation, and you have to mention it's a reduction. The NADPH is reducing it, and the ATP provides energy for it. So that's relatively straightforward in the scheme of things. Using the information in the passage, describe how the structural properties adapt to its function in photosynthesis. So you just mention the photochromatic or photosynthetic pigments, um, which is chlorophyll, which is in chloroplasts in the thylakoids and the granum. And then maybe you have to mention something about it's not the matrix, it's the stroma. You have to mention something about the stroma as well, maybe. So let's see what the answer is. Large surface area for light absorption. Didn't think about that. Layering membrane allows a pigment, yeah. Permeable membranes are diffusion of gas, carbon dioxide, hydrogen acceptors. Okay. So it's also about the membrane we need to remember. Stroma matrix containing enzymes, carbon cycle, yep. Okay. Not too bad. So remember this. It has a large surface area for light absorption. And this. The membrane allows diffusion of gases, carbon dioxide. Oh, and this. Provides surface for attachment of electron, hydrogen acceptors. Okay, I think that's all I'm going to do for this one. It's short, but gone over this topic. It's not a super long topic, so it's good to just get that stuff back in. And yeah, that'll be all for this one.